Let me just remind everybody that it will be recorded. Nina.
Good. So yeah, it is my pleasure today to introduce the ALLODD seminar's speaker, Marc Nazaré. He studied chemistry in Karlsruhe, where he also did his PhD with Professor Waldmann, focusing on organic synthesis. After his PhD, he joined the medicinal chemistry department at Sanofi, where he worked until starting his independent research group at the Leibniz Institute for Molecular Pharmacology in Berlin in Germany in 2013. There, his research focuses on the structure-activity relationships and the multi-parameter optimization of pharmacologically active small molecules, as well as the development of chemical probes for target deconvolution and imaging. And today he will present some case studies of his recent work. We're looking forward to your talk, Marc.
Thank you, Nina, for this kind introduction, and hello, everyone. It's a real pleasure to see some of you again after our fantastic meeting in Barcelona. And today I would like to present you a little bit what we are working on in a kind of snapshot fashion and what approaches we are using for the evolution of chemical probes. And just a small recap, what are chemical tools? They are small drug-like molecules, very similar essentially than drugs. However, we have some criteria not only on the activity, but also on the selectivity of those compounds. They need to be specific, in particular specific to related proteins within the same family, and they need also to exert, of course, a cellular activity. Investigations with chemical tools are mainly performed in cellular, but the big advantage is, of course, that you can translate into tissues, but also into in vivo situation. What you see here is also in part one major feature medicinal chemists have, that they collaborate with drug designers, that they collaborate with biologists, that we take advantage of screening approaches to evolve these hits. That's very important to recognize that medicinal chemistry is really a very interdisciplinary discipline, and in particular also it has a privilege that we can shape the molecules, we can adapt them to a certain need. Just to advocate and also advertise chemical tools for the investigation of target engagement and also mechanism of action studies, we should really recapitulate that it is possible with the same ligand to investigate the mechanism of action and the target engagement also in proteins, in cells, in tissues with the same compound. And one of the main properties which is so advantageous is that it is able to pass any, or hopefully any, biological barrier. It can be used in native cells across species, which is not possible with antibodies, for example. You can attach several tags, cages, fluorophores, and I will show you one of these examples. And of course, these chemical tools are also starting points for drugs. But please be reminded that it might look very boring if we attach perhaps a methyl group at a certain position if we have a kind of a nitrogen dance around a ring system, if we attach a small group at one side. However, this might have a huge impact to affinity, but also selectivity. And I show you here one example, a DPP4 inhibitor, where a single methyl group is crucial to exert really acceptable affinity, or vice versa. If you place a nitrogen at the wrong position, you will lose more or less the affinity. And that's also the phenomena if you consider the dark matter or latent hits within screening collections. Essentially, the message is, if you have a micromolar hit, there's always the possibility to modify this hit to a really highly potent compound. Just the stories which I would like to present you today is a hybridization approach where we use the former semi-optimal chemical tool and merged it with another fragment to a very much improved chemical tool. A second case study will be the generation of active site inhibitors for SHP2. And the third study will present you also the possibilities of a reverse design approach where you convert a drug-like molecule to a fluorescent probe. So first, let me present you how we discovered Tankyrase inhibitors from a phenotypic high content screen. At the FMP, we performed a phenotypic screen based on a Wnt beta-catenin co-localization assay, really target agnostic screen. You may know that the Wnt signaling pathway is very relevant in many cancers and is also responsible in the formation of drug resistance, but also metastasis. And after screening a rather small library of compounds, a follow-up specificity screening and hit validation, our screening unit came up with this compound here, JW74. At that time, it was not known which target this compound had. So a chemical modification attaching a linker for affinity capture MS analysis revealed then Tankyrase 1 and 2 as the prime targets, as the molecular targets of this compound. And Tankyrases at that time were just very new targets with more or less no chemical matter to address. In particular, for specific ligands, there was a shortage in compounds. And what is interesting about Tankyrases is that they belong to a larger family of PARPs, and these enzymes are responsible for in mediating many responses. In particular, the Tankyrases are very important to guide or to prevent the localization of co-localization of beta-catenin into the nucleus, and thereby, by preventing this co-localization, the oncogenic transcription is blocked. If you look at the actual substrate of these Tankyrase here, you can recognize here the nicotinamide and also the adenine core, and you can read a little bit in the pocket architecture how the enzyme might recognize also the substrate, but also a potential ligand. So essentially, this was the hit structure. Had already a decent affinity, but the cellular activity was rather borderline. In addition, the compound was rapidly cleared in liver microsomes, so any investigation in vivo revealing the possibilities of the blockade of Tankyrase in vivo in cancer models was not possible with that compound. So a rather very conservative analog campaign or analoging campaign then generated this compound here and had high affinity, high cellular activity. However, if you look at that compound, it doesn't contain any sp3 hybridized moiety again. And it's a highly conjugated, highly rigid system. And this is really a big drawback, because this compound was not stable under daylight conditions and had bad pharmacokinetics in rats, and showed also some in vivo toxicity findings after multi-dosing. So, and we attributed mainly this bad or rather detrimental characteristics with the highly conjugated system, which was in addition connected to such a double bond, which in many cases also is a Michael acceptor and very reactive moiety. We obtained also a co-crystal structure of this compound and were surprised to observe the binding mode. It accommodates only in the adenosine binding pocket and not in the nicotinamide binding pocket. And you see here co-crystal structure even in the presence of the nicotinamide. So with this feature, this compound was kind of special and a very rewarding lead, because if you look at the pocket architecture, the nicotinamide pocket is really highly conserved over all PARP enzymes, but not the adenosine binding pocket, which is only in this kind of shape present in the Tankyrase 1 and 2 enzyme. And this has consequences. For example, if you use for characterization such a molecule which only addresses the nicotinamide binding pocket, you will end up with a very unspecific response. What you see here is really the selectivity pattern of this compound, and it addresses really, besides the Tankyrase 1 and 2, a lot of other PARP enzymes. Contrast, by addressing only the adenosine binding pocket, you see mainly inhibition only on Tankyrase 1 and Tankyrase 2. So we started for this reason also with this lead, but we wanted to get rid of this highly conjugated system. So essentially we virtually deconstructed this moiety, dissecting the right-hand part. This was done in collaboration with Larry Lötio from the University of Oulu. And then we checked and docked many proposals what to place at this right-hand side. And even while the docking experiments were progressing, there were no really good ideas, good and stable docking poses. But we came across to such a ligand and to this kind of moderately active Tankyrase 2 inhibitors, and we're quite appealed to see that contains more sp3 centers, this aliphatic moiety, which would bridge more or less the double bond, showed up for us as a favorable compound. And in addition, there was a co-crystal structure with this compound, and it nicely overlaid also and aligned also with our ligand. So our merged compound and candidate would look like that here. And what separated us between testing and the design was really the synthesis. It was a 9-step synthesis. And you can see it was more or less efficient with 1% overall yield, particularly because the last steps of the synthesis were really very low yielding initially. So truly, for such a synthesis, you need to believe in your synthetic target very much, and it paid off. As you can see here, we got with one compound already quite decent affinity and kind of micromolar activity in the cellular assay. But a small modification, such a ring contraction here, yielded then rather potent compound also in the cellular assay. And merging another feature, this nitrile group here, to the new compound, improved the affinity or activity, in particular the cellular activity, once again. And within 4 compounds, we really identified a new, very promising chemical tool with high affinity and high cellular potency. And it turned out to be true that this merging was really perfect also in recapitulating the binding mode. If you compare here this right-hand part of the molecule, which is similar to this Amgen compound here, and the left-hand part, which was perfectly matching to the interaction pattern of the parent or former 2 compound. With regard to the profiling of the drug-like properties, affinity, selectivity, we observed more or less a perfect profile. There was no suspected kinase activity. This was a little bit critical as we were concerned about this kind of potential hinge binding moieties here. Permeability, metabolic stability was also acceptable. The only problem for in vivo drug-like properties, if you want so, was the CYP inhibition, and the mouse and rat PK was only moderate. But as a 2 compound, I would say this compound was really, after having synthesized 4 compounds, really a very, very promising lead for further evaluation. And we did so, we checked for this, with this compound in a xenograft model, for example, and we could see really a high efficacy even at lower doses. So the tumor growth was essentially blocked, what is possible via this mechanism by blocking Tankyrase. So it was saturated, the effect, already at rather low doses of 15 milligrams per kilogram. At that stage, we teamed up with Marc to further improve the properties like permeability, but also the PK properties to optimize CYP inhibition. And with this really very robust lead, you could attach many other substituents, as you can see here. 144 modifications were done in a really very efficient way. Of course, the synthetic pathway was a little bit optimized, and many compounds were really of high affinity and potency also in the functional assay. Another kind of investigation I would like to present you now is our concepts towards the optimization of an active site inhibitor for SHP2. SHP2 is a phosphatase, and as you might know, active site inhibitors of phosphatases are usually very complicated to optimize with regard to activity, selectivity, but also cellular permeability. And just a little bit of background on the biological significance of SHP2, although you might have heard quite a bit with the presentation of Nina at our last meeting in Barcelona. SHP2 is a non-receptor tyrosine phosphatase and is involved in the MET and RAF cascade signaling, and it's responsible, or upregulation, overactivation of SHP2 is responsible for tumor formation, cancer metastasis, but also drug resistance formation. SHP2 is usually existing in a close conformation where the active site, the PTP domain, is covered by two SH2 domains. And upon activation, the active site of SHP2 is exposed towards the substrate or is addressable by our active site inhibitor. Quite a while ago, there was a virtual screening performed. At that time, there was no crystal structure of SHP2, so a homology model was designed and used for a virtual screening approach. And this yielded this pyrazolone compound here. This pyrazolone compound, as you might see, was moderately active, however, displayed quite good selectivity. On the other hand, with regard to the chemical features, there were some severe issues. One was that this, let's say, hydrazone moiety was rather unstable. Over time, we observed oxidation. This nitro group is really a toxophore. So essentially for any in vivo investigation, these two moieties would be not very suitable because metabolic activation degradation would occur, and even toxic metabolites would be taken into account. In addition, there was this really highly charged moiety serving as a phosphate mimic, and this prevents usually from passing any biological membrane. Only non-charged molecules, as you know, will pass biological barriers and membranes. So with regard to our strategy, we wanted to definitely change the situation here with this hydrazone. And in addition, we wanted to remove these unwanted fragments, this nitro group here, and also this sulfonate here. And with regard to the rescaffolding, we would consider, for example, by maintaining the decorating vectors, we would consider such scaffolds like indazoles, aza-indazoles, aza-indoles, indoles, and also pyrazolo-triazoles. And just to summarize this quite tedious optimization in 4 compounds, we introduced here such a conformational lock towards these pyrazolo-triazoles with moderate loss of affinity. This was followed then by a rescaffolding towards such a 7-aza-indole, and we more or less recovered the affinity. And by changing then the peripheral substituents and exchanging the unwanted fragments or functional groups, a nitro group, against such a coumarine, and the sulfonate against such a carboxylic acid, we obtained a rather active SHP2 inhibitor. However, and this highlights a little bit the issue of any active site inhibitor for phosphatases. The selectivity is rather moderate for this compound. So within my assessment, I would say this is not a very, very selective and appropriate chemical tool as an active site inhibitors if you want to dissect the role of different phosphatases and contributions versus SHP2. Nonetheless, taking advantage from this intrinsically fluorescent coumarine moiety here could really show how permeable the compound is. You see here are 3D confocal images here at the start. After 20 minutes, you see this bluish colored or stained cytosol, and you see also the co-staining of the cytosol. So it's really well permeating into the cell, in contrast, for example, to the former sulfonate. Furthermore, we could show decent activity in a so-called scatter assay, and this assay is more or less an in cellular model of metastatic outgrowth and migration. Upon stimulation with the hepatocyte growth factor, for example, such pancreatic tumor cells usually scatter and migrate, but you can really block this in the presence of this inhibitor. We could also show with this inhibitor that cellular resistance formation against vemurafenib, a BRAF inhibitor, is blocked, so that vemurafenib still retains its activity in these resistant cells. So, as you might imagine, there's a lot of SAR work in designing these tools. So many compounds have to be made until you achieve a good and very specific tool. Essentially, everything is condensed in one compound. Many data points are hidden and maybe also forgotten once you have your tool compound. And this, of course, this SAR, even if the structure of the protein is not known, you are, with the SAR information, quite confident where you can place a substituent to increase interaction, or where it's detrimental for attaching anything, or vice versa, where there's no change in affinity. And this can be really used for adding a reporter group. And these are, in our understanding, kind of tailored chemical tools or probes, where you adapt your chemical tool towards a specific chemical biology applications. So having the information via the SAR, where to attach a linker to design a PROTAC, to attach a fluorophore, or attach a biotin for a pull down, is really highly relevant. And I would say very often this is forgotten. Nonetheless, we can take advantage of the SAR of many optimized drugs. And this will be the last part of this talk. We converted a very drug-like CB2 antagonist into a fluoroprobe in collaboration with Uwe Greta from Hoffmann-La Roche, and could specifically visualize this CB2 receptor in cellular models and even in vivo with such a probe. So, small recap on cannabinoid receptors, the endocannabinoid system. You might know that the endocannabinoid system is a lipid signaling, and it all roots back to very early chemical biology efforts from Raphael Mechoulam, who deconvoluted essentially the active ingredients of cannabis, discovering cannabidiol, THC, but also enabled others to characterize the entire system, the receptors, the CB1 receptor and the CB2 receptor, discovering also the endogenous ligands 2AG and anandamide. And these efforts, after all, culminated, of course, in the discovery of the CB1 receptor and the CB2 receptor with regard to all the structural information by a crystal structure and a cryo-EM structure. Now, about CB1, much is known. It has mainly a role also in the ZNS system. Much less is known with regard to the CB2 receptor, and there's no drug in the market. This is in contrast to CB1, where there was already a drug approved, but this was shortly after approval withdrawn due to severe side effects like depression and suicidal thoughts. The CB2 receptor is mainly expressed in the periphery and there on immune cells. However, the role is much more unclearer, and it's a kind of chameleon-like receptor with regard to expression, functional activity. And at that time, where we started this program, there were many questions about how to detect and identify the receptor in different tissues, in different cells. There were issues with the translatability of animal models, and there was no specific CB2 antibody at that time. In particular, also, of course, no fluorescent probe to label the receptor. And this Western blot should illustrate you the problem with the antibody. There was no, or there is no specific antibody. And you see in some cells here, you might trace the CB2 receptor, but in the majority of cells here, you might not be able to detect CB2. And in addition, of course, there's no translatability within one humanized antibody or human antibody. So, and there was no mouse antibody at that time. So with our lead here, starting lead should be a rather straightforward task to attach at some region a linker guiding the fluorophore outside of the pocket. However, this is really a severe challenge, as you might see based on this small movie here, illustrating the crystal structure or co-crystal structure with an inverse agonist. The inverse agonist here is really well trapped in the receptor cavity, and there are not too many attachment points where you could attach then a corresponding linker and also respective dye outside at the extracellular domain. In addition, with such fluorescent dyes, you add a large reporter and also a large lipophilic moiety in most cases, or highly charged moiety, which alter the overall properties of the molecule a lot. So with lipophilic dyes, you might observe unspecific binding, for example, in the membrane, or with highly charged dyes, you might see no binding at all. So Thijs Gatsi, nonetheless, took over this challenge, and initially we envisioned to attach our linker at this side. This was based, since no crystal structure at that time was published, based on an homology model. And we tried a very small kind of non-interfering dye initially. And Thijs did a lot of linker variations, but this was really disappointing, as you can see here, with regard to this attachment point. So we observed essentially no activity upon attaching such a linker in this region. After analyzing this a little bit more in detail, we also observed not only complete depletion of affinity, but also the linker attachment point was not stable, and we observed hydrolysis at this attachment point. So we changed again the available lead. That time we were quite courageous, since this compound was not very specific towards the CB1 receptor. However, it should be stable at this region, and we wanted to attach our linker in this region here. And this was really very good selection and highly rewarding. As you can see here, although we lost affinity in binding by attaching here the fluorophore and linker, this still was a nanomolar binder to the CB2 receptor, and the selectivity versus CB1 was even increased. Also very important, the recognition element, or the agonist, and also the construct was still not altering the function as or the functional activity as an agonist. So it still was a highly potent agonist with this fluorophores attached. And what was truly remarkable, we could attach more or less every dye without losing the affinity towards the receptor, and mostly being rather selective versus the CB1 receptor. And this is very unusual. Essentially, you could provide collaborators the free amine, and they click or conjugate the fluorophore of choice for their experiment onto this platform. So we shared this probe with over 25 collaborators, and I will show you briefly in the last 5 minutes a few of the experiments done with this tailored probe. For example, Dimitri and David used this fluorescently labeled probe to design a time-resolved FRET assay using a terbium labeled receptor, CB2 receptor, sorry, measuring the kinetics of the binding and deducing from this equilibrium binding data. For example, you can see this with this silicon rhodamine probe here, and you see essentially over a rather large concentration range, no strong increase of unspecific binding. With this system, they set up an equilibrium competition assay to determine the affinity in a non-radioactive binding assay for non-labeled compounds. And this is in particular important in an academic environment, of course, to have a non-radioactive HTS capable assay. In addition, it's very easy with this assay setup to determine the kinetic parameters like K on, K off. Getting closer to labeling or cellular labeling and imaging, for the first time it was possible with such a fluorescently labeled CB2 agonist to perform FACS experiments. So it was highly sensitive in detecting human CB2 and murine CB2 overexpressing cells. and in addition, very specific versus the human CB1 and also versus the wild type cells. Essentially, we observed no signal for the human CB1 overexpressing cells. Specificity was also shown by using known CB2 ligands, inverse agonists or agonists for blocking this labeling, and you can see this here is observed in a very nice concentration-dependent manner. Now, lastly, some live cell imaging experiments. We also used this cellularly permeable silicon rhodamine dye, which has the big advantage over, for example, any antibody also to be able to stain internal compartments and membranes containing CB2, the CB2 receptor. So in CB1 overexpressing cells, you see almost no staining, a little bit of these red staining you see here, unspecific staining, but you see a very bright signal in confocal imaging studies here for the CB2 overexpressing cells. Now, taking advantage of the cellular permeability of our probe, we could also see accumulation near to the nuclei over time, and we could even follow this via a small kinetic study, if you want so. And in high-resolution live cell microscopy, we were able then to observe the staining of internal membranes, also reminiscent to Golgi endoplasmatic reticulum. We could also stain here. What you see is murine splenocyte native cells, but also human macrophages, all with the same kind of probe. We went even one step further in zebrafish, and there we could see also migrating macrophages here, migrating from the hematopoietic system into the bloodstream. These macrophages were co-stained with M-cherry, so that it is really very well shown that even in zebrafish in vivo, we could use our probe. So one probe platform, around 17 labs who participated in this study in characterizing the probe, but also the CB2 receptor. And I would say this is the kind of way how you should approach always probe work, collaborating, validating the probe independently by other labs, and then, of course, sharing it with all possible collaborators. We are currently working on this topic and extending our studies to the CB1 receptor, but also to the CB2 inverse agonist. As you might know, it's also relevant to label the resting state of the receptor and maybe also dissect, by that mean, receptor pools or populations. In addition, taking advantage of docking experiments and the proximity of several lysines in the extracellular domain of the receptor, we are working on a kind of traceless labeling approach in covalently labeling the receptor and transferring a moiety onto the protein in a kind of non-genetic fashion. This is the work of Leo Mach, Boscha Wajinska, and Anahit Omran. And we hope that we can then trace the receptor by a covalent tag, but having a kind of still free binding site in the receptor to work on the pharmacology with endogenous ligands, with non-labeled ligands. With that, I just want to briefly conclude. I hope I could show you with our Tankyrase inhibitor study how rewarding it might be to consider such a hybridization approach for the rapid generation of a new inhibitor. I showed you how we can quickly get a new, more drug-like scaffold by a rescaffolding approach, and how we nonetheless need to consider unwanted fragments and how to remove them. In addition, a major part of this talk was about the cannabinoid receptor probes, and there I could show you hopefully that it is very rewarding in modifying drugs, profiting a lot from the SAR work, which was formerly invested in designing this drug, and that this is a highly efficient and useful strategy to further adapt this towards chemical biology applications. With that, I would like to close, not without acknowledging, of course, all the members in my group, acknowledging, of course, the funding agencies, and of course, thanking you for your patience and attention. Of course, I'm open for questions if there are any.
Thank you, Marc. Excellent talk, very interesting. I would like to ask everyone to either raise their hands if they have questions or post them in the chat. I see there are already a number of questions in the chat by Zoe. Zoe, would you like to unmute and discuss with Marc, or should I read them?
Yes, sure. Marc, wonderful talk. I really enjoyed it. You showed us all the spectrum, and it was very educational, but also interesting from a scientific point. I really enjoyed it. The concepts were also understandable, I think, by everyone. So I really enjoyed it. Thank you so much for this wonderful webinar. And I do have a couple of questions. So I always read about the probes, and then I always read about the candidate drugs. And I wondered, what is the difference of a candidate drug and of a chemical probe?
Maybe sometimes they do not differ so much, but usually a chemical probe is mainly focused on specificity. If you want to study target engagement and mechanism of action, you really want to have a very selective probe. Think about all the kinase inhibitors, which exert a kinase polypharmacology in most cases, which is wanted for, let's say, treating cancer, which is wanted in many cases to influence in a strong efficacy signaling pathway. However, with the chemical probe, we need to be very strict with the specificity, and that might be the difference. Of course, the drug, if you think about all the safety aspects, we could tolerate, perhaps even for a chemical probe, that it is mutagenic. Yeah,
I see.
but this is not tolerable to have a positive Ames test for a drug development candidate. So
thanks.
all
Yeah,
the secondary aspects with regard to the pharmacokinetics and so on, this is one of the main differences, I would say.
Okay, thank you. I have a couple of other questions. Maybe I will ask one more and then Ai-jen can ask her question. How did you select the libraries to screen for each case? Do you have a standard library for everything, or is it case by case?
So our library is not so large at the FMP, but it's, let's say, not a historic library, but a library which was more or less designed, was a common substructure concept, which was more or less deduced from the approved drugs which were out there at that time, using kind of privileged fragments. And we usually screen them entirely, because we want also the bioannotation by all the targets which are screened. If you screen our library, you will always get also the information if this is a frequent hitter, if it's a kind of a privileged compound for a certain pathway target. So
it's a standard library. They're not custom libraries.
There's no focus library.
Okay, yeah. Thank you. Thank you, Marc. I have a couple of other questions, but I'll let Ai-jen speak now.
Ai-jen, go ahead.
Okay. Thank you, Zoe, for letting me jump in the queue. So firstly, I also concur Zoe's sentiment. So thank you, Marc, for this very clear and informative presentation. I enjoyed the whole presentation a lot. So my questions are specifically about your CB2 fluorescence probe designs. And I noticed that whenever a fluorescence probe is tagged on, you always have a drop of potency compared to the parent compound. And it's, I mean, from the data you show, it seems to be like 10 to 20%. So from your experience, is this acceptable? Or at what level of drop-off should we be concerned about our fluorescence probe design?
I think we always observe this with the receptor. So it is not so much with enzymes. We have another example.
I see. Okay. Yeah.
So it might be a feature of this constellation with this ligand, but however, also with related ligands for CB2, you see a drop. It might be also related with entropic reasons, that there are restrictions for the linker, which needs to be pre-oriented, so that you lose some part of the affinity. But the drop was never that large, as you also noticed. And essentially, we are always at risk to overinterpret the affinity in these membrane preparations, which is also rather artificial. And what we want to see is really a specific labeling at the end. So it was not so much to have the pharmacology behind, which was still, with regard to functional activity, really fine in the single-digit nanomolar efficacy range, but the staining was the most important thing, since most fluorescent probes which were out there for CB2 were super unspecific, diving into the membrane, losing the receptor. Yeah.
Mm-hmm.
Does
Yeah,
this
I
answer it?
definitely noticed that in terms of function, yes, you don't seem to lose as much as for binding. That's certainly our experience as well. In fact, yeah, when we design our fluorescence probes, we always observe a drop off, and roughly at 1 log unit. So which is precisely what you show with your CP2 example. So that's why I was intrigued. Yes. So my second question is, how often do you scan your linker length when you design your fluorescence probe? Is it always straightforward to anticipate how long your linker should be? Do you have a philosophy or design philosophy for linker?
It's rather empirical. So we start really with very short linker and have a kind of modeling assisted linker length range. Yeah,
right.
But this is really empirical, as it is for the design of PROTECs and so on. Yeah,
sure. Okay, thank you very much.
No philosophy behind it.
It's always good to try. So thank you very much.
Thank you.
Thank you, Marco. Then I would ask you to go ahead, and Zoe, maybe we can come back to some more of your questions afterwards.
Okay, thank you. So thank you, Marc, for the very interesting talk. And I have a curiosity about the SHP2 part of your talk. That was the second one. And so you mentioned that your SHP2 inhibitor was not selective relative to phosphatases.
Yeah.
Do you have any idea of how come?
I would love to have a crystal structure to answer this a little bit more and to also optimize the inhibitor, because I believe if we would be more definite with the kind of decoration, we could perhaps gain selectivity. But so far, there's not even a clear picture how the binding mode of this inhibitor is. But I'm a little bit skeptical, honestly, that we can achieve. That's why we go for the allosteric ones. Of course, I'm a little bit skeptical that we can achieve really a strong or a shift over all the shown phosphatases of a factor of 30 so far. Yeah. Without sacrificing too much of other properties.
But so my follow-up question is it known that some of the ligands that bind to SHP2 are also binding to the phosphatases? I mean, do you have this lack of selectivity for other compounds which are known?
Yeah.
Okay.
Yeah. That's an issue. And that was also the kind of major hurdle and the major breakthrough with the allosteric ones. Usually the selectivity was in the same range. And we also didn't manage to improve this significantly. However, we want to give it a try with a covalent inhibitor. Maybe such a handle will help to increase the selectivity, as it is known and shown with kinase inhibitors.
Okay, thank you very much.
Zoe, then I would suggest that we come back to your question.
Yeah, one related question with Agen about the fluorescent probe. How do you know, or what experiments do you perform to know that the probe, the attachment of the fluorescent probe does not alter the dynamics of the process you're trying to monitor?
So that it is really a spectator. Yeah.
That it will have the same effect as the ligand alone.
That's a very good question, because we are concerned on influencing too much the pharmacology. That's why we are interested in the inverse agonist. You know, with the agonist, you are also able to really promote internalization of the receptor. And we were not so sure, for example, if the accumulation in the intracellular compartments was due to the agonist stimulated internalization, because 0.4 micromolar is rather a large concentration. And that was one point which inspired us also to address the resting state. So far, what we can see, there is some effect. Dimitri Leprintejev is investigating this, and I guess he published parts of it. With the linker, we have some bias signaling, of course, if you are referring to that, or shift in the bias signaling, because for sure the kind of extracellular and upper parts of the receptors are perturbed in part. I would say so. It's not, the label is not silent at all.
Okay.
Yeah, that's a drawback. That's why perhaps the traceless labeling would be an escape, hopefully.
That's why the what?
The traceless labeling, where you transfer the label to the protein and leave the binding cavity, or release the ligand from the binding cavity, might be appropriate and give new insight. Yeah.
Okay. Yeah. Thank you. But there are cases where the probe doesn't, the fluorescent tag does not affect, or always there is an effect.
We have the ligand. This is drug-like, and it was designed to have an effect. That means
no, the fluorescent part. I mean the other end.
I mean, with regard to the functional assay and so on, we didn't observe a major difference. But if we look at the beta-arrestin signaling and so on, there is some difference in pharmacology. But as the major aim was to visualize at least the receptor, which was at that time not possible.
Yes,
I think it was a step forward.
Absolutely. No, it's very impressive also. It was just an encyclopedic question too, because I'm not familiar with the probes, the fluorescent probes, I mean. I had another question for slide 16, where it was, I think, the Tankyrase. No, it was the next project, where you showed the optimization of the probe, that you mentioned that you contracted the ring and that brought up the potency. But at the same time, I noticed that you reversed the stereochemistry as well, which, I mean, it's a relatively very big change. So I was wondering about that. Yeah, it was slide 16. So you also reversed the stereochemistry.
Sorry, maybe this was
slide 16. Yeah, you see?
This was, this is just a trans, but it's not chiral.
Okay. But
yeah.
Still... Okay, I see.
Because there is a kind of axis. But the interesting point of your you spotted an aspect which is really interesting. You observed here a drop isomerism, because the rotation here is hindered.
That's right. Yeah.
We discovered this by chiral HPLC later on, and with the fluorine you can separate enantiomers. With the fluorine you cannot separate anymore. So this is maybe escaping the attention very often. Also, old drugs sometimes have this kind of drop isomerism.
Hmm.
But with the free rotation of this lower phenyl ring, there's no chirality in the molecule.
Okay, yeah,
You
but
only have to be anti.
right. But yeah, I think so. In the case, in the in the first lead, basically you have a restricted rotation of the ring, and in the next one with the contracted ring, you have a free rotation.
No, still not.
No.
Still not. You could isolate this, and the melting temperature was, if you want to call this melting temperature, 70 degrees. So really stable at room temperature. Yeah, you could separate the enantiomers, and only with the smaller fluorine, there was free rotation at room temperature.
I see. Okay.
Yeah.
And this also plays a role because this chlorine is important for binding it to be in this position. I see. That's very, very interesting.
Yeah.
Okay. And then about the replacement of the Michael acceptor, are there known strategies to replace them, or you have to go case by case?
At least eliminating the double bond. Sometimes it works with a three-membered ring, but the four-membered ring is also on the way to replace it. Yeah. Essentially, that would be the major strategy. Sometimes you can freeze by substituents a little bit the conformation. But essentially, in this context, conjugation, aromatic system here and here, this is not a good idea.
I see. Yeah, that makes sense. And then two more encyclopedic questions for the. It was interesting that they had this retraction of the CB1 approved antagonist because it caused suicidal thoughts, and I was wondering what was the off target, to which protein did the CB1 antagonist bind to cause the suicidal thoughts?
Yeah. So I would say THC consumers, this is really chemist interpretation.
Okay.
For the agonist THC, I would claim that most people are happy and relaxed, right? Cannabis consumers. Yeah, and with the antagonist, it is vice versa, but this is only part of the story. Very recent data show that it is an interference, I think, with interleukin-1. And by the way, this is a pathway which is shown now for some ZNS drugs, because this is not the only ZNS drug which has as a side effect suicidal thoughts. This is also a component of some of the antidepressant drugs, not for all. And I cannot unravel the entire investigation now, but I can refer to some of the results. So it's really now possible with a kind of bioassay to characterize those drugs and recognize them much earlier than before. Yeah, on a biochemical level. It has to do with serotonin receptors and so on expression.
Very interesting.
So the actual target inducing this is not known for most drugs, but at least you can detect this much earlier than before.
But in their case, they couldn't have known it or tested beforehand about it.
So there were already suspects in clinical phase 2, all people who had already depressive episodes and so on were excluded from the study, and nonetheless, this was observed. So it's a very significant effect and side effect of the CD2 agonist, and a related antagonist, sorry, a related CB1 antagonist also showed this.
Mm-hmm. Very interesting. And finally, I have a more practical question, because I was very impressed that you showed the 25 collaborators with which you shared the probe. And I was wondering, since you patent, I noticed in the beginning you mentioned something about patents, and I believe this chemical matter is patented. I was wondering on which grounds did you share the probe with your 25 collaborators? Is it a non-exclusive license, or do you expect royalties from them, or some kind of other in-kind contributions?
That would destroy any kind of collaborative network. For any research purpose, we share this. And actually, this probe is not even patented, and it shouldn't prevent any kind of academic collaboration, any research attempt. You just need to drop me an email with regard to any of those probes. We will see how much we have. That's the only hurdle to share it. Yeah. No.
But the question is, I mean, you showed us very exciting stories, and my actual question I was hinting towards, like, how do you decide which ones to share with the community and which ones are more could be marketed, or you don't care about that, you just share everything?
So with regard to the probes, we of course share this, and our prime aim is to publish. Any patent will not prevent us from publishing. And if we have a patent, for example, on a certain probe, it's even easier to share it with the community. You know, any kind of development path, for example, with the Tankyrase inhibitors, is not precluded then. And usually this is no issue at all, because paperwork usually prevents most of the collaborations.
But I mean, if you patent, then after some point, the patent becomes very expensive, and you have to pay 30,000 euros per year or something in fees. Like, shouldn't this patent be also bringing in some? What is the reason for patenting then? Yeah, with regard to the example, for example, the SHP2 inhibitor, we didn't. Yeah. The first example, I think you patented the Tankyrase inhibitor was patented because there was a clear path also towards drug development. There was then a CRO, Merck involved at that point. And this is really then an expensive program, and all the tech transfer offices request also a patent. I see. At that point. Okay. It's also one of the elements to get certain funding for such translational projects. Absolutely, I agree. But thanks for this is a great insight that you shared with us. So thank you very much. I don't have any more questions. So sorry about the many questions, but I just found everything so fascinating. Thank you so much. Thank you.
Thank you. Also from my side, are there any more questions from the audience? If not, I would like to thank you again, Marc, for this insightful presentation, and wish everyone a nice afternoon. And I guess we'll see each other again for another journal club and another webinar before Christmas. So Until then, thank you very much.
Thank
Bye,
you.
everyone.
Thank you for the
Goodbye.
presentation.
Bye
Bye.
bye.
Thank you. Bye.
Thank you. Bye.